In this video, we will learn about enzymes inhibitor. But first, we need to know what are inhibitors, their types, and their function. Inhibitors are low molecular weight chemical compounds. They can reduce or completely inhibit the enzyme catalytic activity. Effects of inhibitor can be reversible or irreversible depending on the bond they make with the enzyme. If enzyme binds with the inhibitor instead of substrate, then enzyme inhibitor complex will form and no product will be generated. Inhibitors are of two types, reversible inhibitor and irreversible inhibitor. As the name shows, the action of reversible inhibitor can be reversed. Their action is reversible because they make weak bond with the enzyme. They bind to enzyme with a non-covalent bond to form enzyme inhibitor complex. Irreversible inhibitor makes strong covalent bond with the enzyme to form enzyme inhibitor complex. Effects of these inhibitors cannot be reversed. Next, we will discuss about types of enzyme inhibition. It is of three types: reversible inhibition, irreversible inhibition, and allosteric inhibition. Reversible inhibition is further of three types: competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. First, we will learn about irreversible type of inhibition. In this type of inhibition, a reversible inhibitor makes covalent bond with the enzyme. Due to this covalent bond, they require high amount of energy to get detached from the active site of enzyme, which is not possible. Most of irreversible inhibitors are toxic substances. Irreversible inhibition. In irreversible inhibition, inhibitors make weak non-covalent bond with the enzyme. Let's discuss its three types. Competitive inhibition. In competitive inhibition, structure of inhibitor is similar to the substrate. Therefore, inhibitor compete with the substrate to bind with the enzyme. This is the reason it is called competitive inhibition. Molecular structure of substrate and inhibitor is also similar. Normally, enzyme combine with the substrate and form product, but here, inhibitor similar in structure with the substrate binds with the enzyme active site, leaving no space for the substrate. No product will be formed and enzyme catalyze reaction is decreased. In competitive inhibition, Vmax of the reaction is not changed, but Km is increased. Some examples of competitive inhibition are: the enzyme succinic dehydrogenase binds with the substrate succinic acid and form product, but its activity is reduced with the help of competitive inhibitor molecule malonate or oxaloacetate. And other example is inhibition of folic acid synthesis by sulfur drugs in bacteria. Non-competitive inhibition. Competitive inhibition is also known as mixed inhibition. In this, inhibitor do not compete with the substrate because it attached on the allosteric site of enzyme. After binding with the enzyme allosteric site, it changes enzyme shape, so substrate cannot identify and bind to the enzyme. Non-competitive inhibitors bind to either enzyme or with the enzyme substrate complex because it do not attach on the active site of enzyme. In non-competitive inhibition, Vmax of the reaction is reduced but Km remains same. Its example is inhibition of enzyme cytochrome oxidase by the non-competitive inhibitor cyanide. This enzyme is present in mitochondria and take part in electron transport chain, which is process of oxygen consumption and ATP production. When this enzyme is inhibited by the non-competitive inhibitor cyanide, no ATP will produce, which ultimately leads to animal death. Uncompetitive inhibition. In this, inhibitor binds only to the enzyme substrate complex. In this reaction, both Vmax and Km will decrease. Other type of inhibition is allosteric inhibition. It is also known as feedback inhibition because it controls the enzymatic activity in a fair way. This inhibition is responsible for normal functioning and maintenance of our body. It works as a feedback loop. Downstream product regulates the speed of upstream reaction. Thanks for watching. Please do like, share and subscribe.